Hi everyone, Miss Longley here, and today we are going to be learning about using codon charts in transcription and translation, which I like to call TNT. We're going to be covering objective number 7 today, which is use the genetic code to predict the sequence of amino acids that will result from a given DNA sequence. So you've been learning about the processes for the last couple of days. Now you're actually going to try your hand at it. So we're going to start with transcription first, since that is the first step. Remember, transcription happens inside the nucleus. We are making an mRNA copy of the gene that is found in DNA. So here we have a sample DNA strand. Most of the time, these DNA strands are like hundreds of nucleotides long. This one is a lot shorter, just for the time being. We're not going to be doing 100 nucleotide bases. So for this one, what I want you to do is take a look at each base. That's referring to the nitrogen base, the letters. And we are going to use the complementary base pair rule to figure out what would the mRNA strand look like. So G continues to pair with the letter C and vice versa. But the letter A is no longer pairing with T because thymine is not found in mRNA. It's only found in DNA. So we're going to use the substitute nitrogen base, uracil, the letter U, in its place. So let's see what that looks like. So for this first one, I'm going to start all the way to the left and work my way to the right, just like reading a sentence. So if I see this base, T, I know that T pairs with A. So that is going to be my first base of the mRNA strand. Here I say the letter A in DNA, and I know that A normally pairs with T, but I'm not going to find T in mRNA. So instead, I'm going to pair it with the letter U, the uracil. And I'm going to keep going along, pairing the complementary base pairs to figure out the bases for mRNA strand. So here we go. G, C, U, C, C, G, and lastly, A. So there's my mRNA strand. I just transcribed it. Now it's your turn. Go ahead and turn to page 11 in your packet, and you're going to try your hand at figuring out what would be the mRNA strand with the given DNA strand below. Pause the video, come back, and check your answer. Great job! Did you get this that you see below? Compare your work. If there's any mistakes, try to go back and figure out where you made that mistake in. Remember, you gotta use the base pair ruling. A pairs with U, T pairs with A, and G and C continue to pair with each other. So the U can get a little tricky, but with some practice, you'll get there. All right, now we're on to step 2. So step 2 is translation. This is where the ribosome is going to be reading the mRNA that just left the nucleus. And the ribosome is going to be linking the amino acids together. tRNA, another molecule, is responsible for delivering those amino acids to the ribosome to make that protein. So let's try translation by hand. So here is our mRNA strand that we previously just transcribed. Now we're going to translate it. In this example, you are kind of pretending that you are tRNA in this case. So we're determining what kind of amino acid should we bring to the ribosome to make this protein. And how to do this is we have to follow the rules of codons. So in the mRNA strand, codons are represented by the 3 nucleotides grouped together. That will produce one amino acid. So here the base is AUG, that is one codon, and it's going to code for one amino acid. If we're to continue counting, so CUC, that's a second codon. And then CGA, that's the third codon. We're starting with AUG because that is considered the start codon. This tells the ribosome where to start reading. It's kind of like the capital letter of a sentence. That's where you know where to start reading a sentence. So go ahead and turn to page 2. With translation, we got to use a little help using a genetic codon chart. So we're actually going to learn about 2 versions, and then you get to decide which one you like better to use. So The first one is called the bullseye codon chart. You'll see that it has the bases listed kind of in a circular pattern. And on the very outside, we have the amino acids. The second type of chart, a little different, but does the same job. This one uses the bases first base, second base, and third base. And then using kind of similar to like reading a multiplication chart, you're going to figure out what the amino acid is. Again, you get to choose. I'm going to teach you both, but then by the end of this video, maybe you'll have a good idea of which one you prefer. All right, so let's translate this codon. We have AUG. So how to use the bullseye codon chart, what we're going to do here is we are going to start with the first base, which is A, and we are going to find it in the bullseye. So with the bullseye method, you always start in the bullseye, in the middle. So here I start in the middle, and I know my first base is the letter A. So I move just a little bit down to the lower left where the A is. Second base is U. So I move a little bit further out to the outside, and that's where the U is. Third base is G. So I move a little bit more to the outside of that circle. And right here, I can see that the amino acid is methionine. So the codon, AUG, is telling me that the amino acid methionine is the correct amino acid that matches. So again, tRNA in this instance would be kind of like the delivery person. So it's delivering the amino acid methionine to the ribosome. You are kind of like tRNA in this case. All right, how to use the codon table chart. We're going to use the same mRNA strand, AUG, but this one's a little bit different. It's very similar to a multiplication table. So I'm going to start with my first base here, and there's the first base in the table. I go down to the letter A. My second base is the letter U, so I go down to U. Notice how they kind of match together. And then my third base is G, which is right here. And so G tells me that it is start. You might be like, what is a start amino acid? That doesn't exist. Start means start codon. So think about start is really just saying methionine. So that's the amino acid that's going to be coded for AUG. And again, it's tRNA that delivers this amino acid to the ribosome in real life. All right, so let's finish this translation example together. We already figured out the first codon. You're going to figure out codon 2 and codon 3. So let's start with 2. So we have CUC. Using your preferred codon chart, go ahead and decide what amino acid is it coding for. Did you get leucine? Good job. Leucine is the amino acid that matches with this codon, CUC. All right, try the third one. Did you get arginine? Nice job. Arginine is the amino acid that matches with CGA. Awesome. That's translation. All right, one last thing about translation is when you get to a stop codon. So we have a start codon, which tells the ribosome to start reading. And then we have 3 different combinations of stop codons, which tells the ribosome to stop, meaning that stop delivering new amino acids. We are done. We're ready to fold up that protein and put it in a vesicle to ship it where it needs to go. So your turn. You're going to turn to page 12 in your packet, and you are going to translate this mRNA strand into amino acids. Come back to the video and check your work. All right, did you get the following below? Methionine, proline, valine, and the stop codon by UGA? Great job. If you didn't, go back and check your work. Maybe you were using a different method. Try a different chart. It's good practice to try both and see which one you really are comfortable with. All right, so one last thing about stop codons. It acts like the end of a sentence, and it tells the ribosome to stop reading the mRNA. So when you're translating, you do the same thing. You stop when you get to a stop codon. All right, let's do example 1 together. So in your Google Doc that you have open, you found this video. You are going to do both steps, transcription and translation. So here you have a DNA strand. go ahead and find the mRNA strand using the complementary base pair rules. Don't forget T is replaced by the letter U. And then use your preferred codon chart to identify the amino acids. Pause the video and check your work. All right, did you get the following below? So did you get methionine, proline, isoleucine, and a stop codon? Great job. If you didn't, go back and check to see where you may have made your mistake. The last part that I want you to do is go ahead and answer these 3 questions. Number 2, I really want to put emphasis on when you're using the genetic code chart, whether it's the bullseye or the table, you need to know the codons of the mRNA. We're kind of disregarding the tRNA anticodons because that's not what we need to figure out the amino acid using the charts. But anticodons are necessary in real life when the cell needs to know what kind of amino acid needs to be delivered to the ribosome. All right, and the last thing that I want you to do after you answer those 3 questions is you're going to finish the remaining 4 examples of transcription and translation in your Google Doc. That is today's ABA. Thank you for watching, and I hope you have a wonderful rest of your day.